Hello class, I'd like to give you a brief lecture on skeletal muscle. Of course, this video is to augment the extensive lecture that I've already started to give you in class and will continue to give you. There's a lot here, and again, for my anatomy students, I am expected to teach you skeletal muscle physiology. So let's start first with this picture of Arnold. Clearly, he's not as fit as he was in the beginning with lots of skeletal muscle mass, but I mean, he's in his 70s. I would argue he still looks pretty good. Let's not debate over his political actions, shall we? First of all, skeletal muscle, of course you all already know that it's very important to maintain your posture. It works across joints and it helps for movement, but it also is a form or is an organ that we use to communicate to others. It's a form of communication. We use our facial muscles, and without speaking words, when I look out at the classroom, I can definitely tell who is annoyed with me, who's scowling at me, throwing the eye darts, especially after a lab exam. So it's very clear when I look out at my students exactly what you're thinking. There's also support of abdominal organs. If you have more abdominal tone, you are using some of your abdominal muscles, in particular like your transverse abdominis, to help support your abdominal organs. And then let's face it, you use skeletal muscle to also control your bodily openings. You certainly can pucker your lips, you can close your mouth, but that is not the only sphincter that you have control over, and you and I both know exactly what I'm talking about. So I just covered the function of muscles. I'm now going to go through the properties of muscle, classes of muscles, levels of muscle structure. You're going to learn a whole set of F words. Don't worry, it is G-rated. And then you're going to see a review of some of the content I've already started to give you in class, where I told you that every cell in your body is set up like a battery. Remember, separation of charge, separation of chemicals. And then we're going to talk about the actual action of muscle contraction and what's required then for muscle relaxation. And then I'll finish up with the definition of motor units, types of fibers, are they slow, white, red, fast, how do we combine those words, and then your lever systems. So let's cover the properties of muscle. Muscle is excitable. That means it can generate an electrical current. Remember, I told you in class that the movement of charge, things that are positively charged, things that are negatively charged, when they move across the cell membrane, they carry with them a charge, and that's like an electrical current. Very similar to when you plug something in the wall and you flip the switch. I asked you in class, would you want to touch a bare wire when the switch was on? And you all said no, no. And rightly so, because you would become, quote, electrocuted. Well, it's a flow of charges, and your cells are capable of doing the same thing as long as they are able to permit flow of charge. And skeletal muscle is capable of doing that. It's conductive, which means the electrical signal can spread across the entire muscle. It can spread. Contractility, it has the ability to contract or shorten. Flex your biceps brachii muscle right now. Flex your lower forearm. In order to do that, you're contracting your biceps brachii, and it's shortening in its length. That's contractility. You also, many of you go to yoga, and they focus on a lot of positioning and as well as stretching. And so you stretch your muscles, and you don't leave your yoga class dragging your fingernails on the ground because you've stretched your muscles and they haven't recoiled. So they have the ability to stretch, which is extensibility, but also recoil back to their original position, which is a very important property of elasticity. There are three main classes of muscle, and you are expected to know all of their properties. both physical, meaning what they look like, as well as functional, meaning how they work. So first of all, again, what they look like, that's the histological appearance. And then the functional classes or functional categories, which alludes to how they work, what makes them contract, etc. So skeletal is certainly our focus for this lecture. It is striated. What does that mean? It means it has a lined appearance. Look at your paper that you might be taking notes on. Is it striated? That means, is it lined paper? Or is it non-striated? Are you actually pulling a piece of paper from your computer printer and it's blank? So striated, there's a lined appearance to it. It's voluntary. That means that you have conscientious control over it. And we're going to talk about these myofibers. These are actually the cells. Is it branched? Are they, what shape do they have, for example? And I need to cover that. Cardiac cells, cardiac muscle. It's striated like skeletal, but it is not voluntary. Do you consciously think about having your heart muscle contract? No, you don't. The fibers, the myofibers, the actual cells are branched. So they are not parallel like the skeletal muscle myofibers. They branch like our freeway system. And we give a special name to the myofibers. Fiber means cell. Myo means skeletal, or I should say muscle cell. In the case of skeletal muscle, the myofibers are parallel in arrangement. In the case of cardiac muscle, the myofibers are branched, and we also refer to myofibers or cells as cardiocytes, the functional unit of the heart tissue. And lastly, we have smooth muscle, which is shown here in the bottom picture of the three pictures that I have. It is non-striated, no lined appearance. Unlike the heart picture that I show you and the skeletal muscle picture, you see lines in the pictures that are drawn for you. Smooth is not lined, it's non-striated. It's involuntary. Smooth muscle lines blood vessels. It lines your intestinal tract. And none of you think, okay, right now I'd like to vasoconstrict my brachial artery in my arm. You have no control over that. You have no control over propelling more of your digestive food more quickly through your digestive tract. You have no conscientious control over that. It's fusiform in shape. That means spindled shape. That's in striking contrast to the cardiac myofibers or cardiocytes that are branched, and also in striking contrast to the myofibers in skeletal muscle, which are more parallel in fashion. Your textbook, doesn't matter which textbook you're using, your textbook definitely will have a section where it talks about the different three categories of muscle, and it will compare and contrast how they're similar and how they're different. For those of you in my class where McKinley is our textbook, but again, it doesn't matter. All anatomy textbooks have this kind of comparison and contrast section. You should definitely pay attention to the size of the myofibers. Are they parallel? Are they branched? Are they fusiform? Is it voluntary, involuntary? Is it, where do you find the mitochondria? Are they squished off to the side? Where do you find the nucleus? Are they squished off to the side, or are they more in the body of the muscle cell? You should know these characteristics. We will definitely test you on these characteristics for this lecture unit as well as for your lab unit 2. So muscle terms. We need to talk about prefixes and where it applies suffixes. So if you see the prefix myo or sarco, you should be thinking muscle. And I can combine myo and sarco with different suffixes. For example, sarcolemma. Sarcolemma means the muscle cell membrane. It's a cell membrane around a muscle cell. I can use myoblast, as I show in this picture, and that should mean these are immature, determined, but not differentiated cells that will differentiate into some type of muscle cell. Again, these suffixes like blasts or sites, that should not be new to you. We should know what those words mean at this point. All we're doing is changing out a prefix. In the case of myoblast or myocyte, you should know that a myoblast is a determined but not differentiated cell that is determined to become a muscle cell. A myocyte would now infer that you are now looking at a well-differentiated muscle cell. To get more muscle cells, when we're talking about skeletal muscle, we don't really have more myoblasts, mitotically active, generating more mature muscle cells. You don't get hyperplasia. That means more cell production. Instead, the cells you have get bigger. That means hypertrophy. Cells that you already have get bigger. They increase in girth, width, and that means they're packed with more muscle contractile proteins, which I will introduce to you in just a moment. You do have some ability for your myoblasts to act as stem cells and could potentially replace any damaged mature skeletal muscle cells. But your ability to do this decreases with age. We oftentimes hear of how resilient children are, and to put that in perspective both physically and mentally, let's focus just for this lecture on the physical resiliency of children. They can rebound from any sort of trauma so much more quickly than our elderly. Why is that? They have time on their side. They have still some residual stem cells that could replace damaged, mature, differentiated post-mitotic cells. Myoblasts are an example. So I have had a cousin in the past who had a severe off-roading tragedy when he was very, very young. His ATV kind of flipped over, and it basically carved out some of his calf muscle. He just completely lost it. You can almost think of it like this ATV, just like a shark, just tore out his muscle. He lost it. But I'm here to tell you that that boy now, yes, you can see some deformity in his calf, but he's got a lot of strength. That muscle has definitely rebuilt itself. Why? His injury occurred when he was really, really young. I mean, 5, 6, something like that. And he still had enough myoblasts to repopulate that skeletal muscle. It didn't come back fully, but he at least regained some muscle mass because of hyperplasia. New skeletal muscle cells populating that muscle. Very, very interesting. As we age, we lose our myoblasts. We don't have as many, and therefore we don't recover from muscle trauma as well. The gross anatomy of muscle. Okay, so we need to talk about your F words, and we also need to talk about wrappings. I sort of like to think of this as when we're freezing food. How do you freeze food? Do you take food, say chicken, in its original styrofoam container with the plastic wrap over it? Do you then put that in a big Ziploc baggie as an extra protective layer? Or maybe you might wrap it in foil first and then a Ziploc baggie. So I want you to think of wrappings around muscle. And I want you to think of how you can take muscle like chicken meat or steak and wrap it with different layers to store it. That's what I want you to think of. Okay, so if we were to go from the most internal to external layer, internal to external, then we would have to start from the bottom of this slide and work our way up the bullet points. So immediately around a single muscle cell, we have endomysium. And then we have packages of muscle cells. Those are called fascicles. And we have another wrapper around this bundle of muscle cells referred to as an individual muscle cell is referred to as a fiber. So a bundle of fibers is a fascicle. And the wrapping around a fascicle is a perimysium. And then you have the outermost wrapping around an entire muscle. That's epimysium. And it's continuous with the tendon for that muscle. And then you have another layer around that called the deep fascia. And basically that's a wrapper to separate one muscle from a neighboring muscle. And then lastly, for muscles that are close to your skin, there's another wrapper to separate your dermis, or I should say your hypodermis, from the muscle deep to it. So that's called your superficial fascia. And we will definitely see superficial fascia and deep fascia when we skin the cats this week. So how can you think of these wrappers and compartments? Okay, let's say you are a smoker and you buy a carton of cigarettes. I want you to think of the carton you buy. If you've never purchased a carton of cigarettes or looked at one in the grocery store, a carton of cigarettes, when you buy it, it's actually wrapped with like a saran wrap. And you have to unwrap the plastic wrap before you get to the carton. I want you to think of the saran wrap that you have to the plastic wrap that you have to take off as the deep fascia. And then finally you get to the cardboard box itself. And you have to open that to get to the individual packets of cigarettes. So I want you to think of the cardboard box as the epimysium, the wrapping around the muscle itself. And I want you to think of the carton around all these individual packets as the wrapping around a muscle. And then I want you to think about taking out a package of cigarettes from that carton. And you, again, have to unwrap the wrappers around from that carton. There's even a plastic wrap and a cardboard container. And I want you to think of the cigarettes in that little individual cardboard box as a group of cigarettes. And the packaging around the group, the carton and the wrapper, I want you to think of that as perimysium. And a single package of cigarettes, I want you to think of that as a group of skeletal muscles, which is called a fascicle. So instead of a package, think fascicle. And then if you were to pick out a single cigarette from that package, one, I want you to think about the paper wrapper around the tobacco, and that's the endomysium. That's what I want you to think of. There are some terms in anatomy for the analysis of muscle, gross anatomy. That means the visual. And that's shown here on this picture. Clearly, muscles have to attach to bones on either end. You're seeing the biceps brachii, and clearly it's attached. It's attached to the humerus, it's attached to the scapula, you're seeing in this picture, and the radius. Now, when muscle contracts, it's going to pull its insertion toward its origin. The biceps brachii you're seeing here inserts on the radial tuberosity. And so when your biceps brachii contracts, you flex your forearm. And the origin of the biceps brachii, you're seeing that the muscle splits into two heads, and it's going to have two origins. That's why bi means two, biceps. That means two origins. An action is the action a muscle will induce across a joint. In this case, in this picture, flexion of the forearm. It's working across your elbow joint. When we designate a muscle as being the prime mover or agonist, we're talking about, hey, when you flex your forearm, the muscle mostly responsible for that, the prime mover, is your biceps brachii. But that is not the only muscle in your arm that can cause flexion of your forearm. There are others that could also help the biceps brachii. Those muscles that help do that action would be called a synergist. And some muscles can help a prime mover do its job better by holding steady a joint to keep it more stable while a muscle is changing the angle of that joint. So synergists and fixators help a prime mover do its job better. If you're talking about muscles that are antagonistic, that means they have opposing actions across a joint. Can you tell me the antagonistic muscle to the biceps brachii, a muscle that causes extension of your forearm? You're right. That would be the triceps brachii, and it's found on the posterior compartment of your arm. It's found on an opposite side of the bones, so it can pull on the bones differently. That's an important concept to keep in mind as we go through this lecture and Lab Unit 2. How are muscles named? They're named in a variety of ways, and I urge my students to appreciate all of these different ways of naming muscles. We can name them for the direction of their fascicles, that means groups of cells. Are they parallel? Are they straight? The word for straight is rectus. Are they at an oblique or slanted orientation? We would say oblique, like your external oblique muscle. Where are they found? Biceps brachii. Brachii means it's in your brachium. That's your arm, you know, where your humerus is. Number of origins. Biceps brachii. Again, bi, two, meaning two heads, two origins. Shape and size. Gluteus maximus, for example. Gluteus minimus. That would refer to size. Or shape. Let's see, what's one up here that has for shape? Quadratus femoris. Quad, meaning it's like a four-sided muscle. Origin and insertion. If a muscle is named for origin and insertion, the origin always, 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 always comes first. So in the case of origin and insertion, we can think of sternohyoid or sternothioid, and those two muscles are telling you origin and insertion. Sternocleidomastoid is shown on this screen. Origin is from the sternum, and insertion is clavicle, and also the mastoid process. Sorry, let me rephrase that. Sterno meaning from the sternum, origin. Cleido, origin from clavicle. That's the part I messed up on. And the insertion is in the mastoid process. Action. Flexor carpi radialis. Flexor. That word tells you it's going to cause flexion. Carpi. Carpus is your hand. Flexion of the hand. Extensor hallucis longus. You know your hallux is your thumb. Extensor, meaning it's going to extend your thumb. So please appreciate the names of all the muscles you will be abused with, because their names are meant to reduce the amount of memorization that you do. Their names tell you a lot about that muscle. So here we go. Now we talk about your F words. What is a fiber? A fiber, regardless of what you're talking about, a fiber, when we're talking about muscle or neurons, a fiber refers to an individual cell. So if I say a myofiber, I'm talking about an individual muscle cell, whether that means a cardiac muscle cell or skeletal. Now, a bundle of muscle cells is a fascicle. Think of that as a package of cigarettes within a bigger carton. Now, let's think of a single muscle cell and peel away the endomysium. Let's peel it away, and let's also peel away the actual sarcolemma, the cell membrane. Peel that away. And if I peel that away inside a skeletal muscle, shown here in the third picture down, if I could peer inside the cytoplasm, I would see rods of proteins. Proteins that are parallel in their organization. Much like the tobacco, if we were to peel off the wrapper around a cigarette, the tobacco might be arranged in a parallel fashion. Then that rod of protein inside a single muscle cell is called a myofibril. And myofibrils in this picture, they look striated. So we're looking at a parallel arrangement of smaller proteins in this rod or complex of proteins. And those arrangements of smaller proteins, the actual proteins, are called myofilaments. And this is where we have discussion of thin filaments versus thick. Thick filaments are made of myosin, and thin filaments, they're a trifecta of 3 different proteins like actin, troponin, and tropomyosin. So again, here is a more up-close picture of an individual muscle cell. Notice that the cell membrane is peeled back, and I'm seeing these rod-shaped arrangements of proteins, protein rods, and one of them is pulled out. And you're seeing that that large protein rod is called a myofibril, and it's made of smaller protein rods called myofilaments. These are your thick or thin filaments. But I also want you to see that the cell membrane is kind of tucked in around all of these individual myofibrils. And where the cell membrane invaginates, well, that's called a T-tubule. I sort of like to think of it as when you're chewing bubble gum and you're getting your gum flattened so that you can blow a bubble, you protrude your tongue gently through that flattened piece of bubble gum, and then you quickly pull your tongue out and you blow air in that little pocket, don't you? So I want you to think of that little pocket that you made like a T-tubule, a pocket that goes deeper into the cell and around the myofibrils. That's a T-tubule. The cytoplasm that's inside a muscle cell, well, that's called the sarcoplasm. That's cytoplasm of a muscle cell. And a muscle cell has endoplasmic reticulum as well, but when it's in a muscle, we call it sarcoplasmic reticulum. And I want you to see that in the kind of blue-purple part of this, you're seeing a bluish kind of web inside the muscle cell. And you're seeing that this bluish kind of web kind of goes parallel to the green picture, the green structure in this picture. The green structure is a T-tubule. Notice on either side you have these swollen ends of the sarcoplasmic reticulum. That's the blue net-like structure. So you have the swollen ends of a sarcoplasmic reticulum on either side of a T-tubule. This creates a triad. Triad means 3. And I tell my students, think of 3 T's together. You see kind of that bluish swollen ending, the terminal cisternae, that's what that's called, the swollen ending of the sarcoplasmic reticulum, right next to a green, and then you see another swollen ending. So you see a terminal cisternae, T-tubule, and another terminal cisternae, T, 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 3 T's, that's your triad. And this functional contact between the terminal cisternae of the sarcoplasmic reticulum and the T-tubule is really important because the action potential, the electrical current that's generated in the cell, remember, it has conductivity, it can spread. It's going to spread down the T-tubules, and that electrical current is going to directly stimulate the terminal cisternae, the sarcoplasmic reticulum, and that's what's going to cause calcium voltage-gated channels to open and release calcium into the cytoplasm from the sarcoplasmic reticulum. This is our last slide for the first segment, and it's a up-close picture showing your proteins that are involved in the myofilaments within a myofibril. So again, thick filaments, down here in the middle picture, you're seeing an individual myosin molecule, and you're noticing that it has two heads and a tail. Well, a single myosin molecule does not make a thick filament. If you look in part B of this middle picture, you're seeing thick filament where many myosin molecules arrange like golf clubs in a bag with their heads radiating outward. Now you've got a thick filament. And these myosin heads, they have an ATPase activity, which means they're going to cleave ATP and release energy, and that energy is required for the muscle contraction. A thin filament consists of troponin, tropomyosin, and actin. And when I see you in class, I will relate an old TV show called All in the Family to thin filaments. And I'll tell you about Gloria and her mom and dad and how protective they were about her. So in my story, Gloria is going to be the role of actin, and her mom is going to be playing the role of troponin, and her father, Archie, is going to play the role of tropomyosin. He's very protective, and if you're looking in this top picture, you're seeing that tropomyosin in green kind of covers over actin, protecting it. And the troponin is going to convince the tropomyosin to shift and change positions and expose actin so that the myosin heads can interact with actin and lead to muscle contraction.